Hello viewers, welcome to my YouTube channel. In this video, which is a part of my video series on biomolecules, I wish to present before you an overview of carbohydrate metabolism. So first let me remind you that carbohydrates perform a number of roles in living organisms. First of which is that they are structural molecules. As you know that cellulose form the cell wall of plant cells and chitin form the exoskeleton of insect. Besides that, they are present as pentose sugars in DNA and RNA also. So this is their first role, which is a minor one. The second role is that they can be used as storage foods. When they are stored in plants, they are used in the form of starch, and in animals they are used in the form of glycogen, and both are structurally related, as I have explained earlier also. Then the most important role for carbohydrates is that they are energy providers, and in which the role of glucose is most prominent. So this is the prime energy providers in almost all organisms. Now talking about the origin of carbohydrates. So mainly carbohydrates come from the activity of photosynthesis, in which plants synthesize glucose from carbon dioxide and water in the presence of light, and you see this is the reaction of photosynthesis through which most of the glucose that we see around us in living organisms is formed. Now this glucose is consumed for energy production through the process of respiration. Whenever animals consume plants, so they take glucose and they oxidize it either through anaerobic method, in which glucose is converted to ethyl alcohol and CO2 yielding to ATPs, or it is oxidized through aerobic respiration, which is a longer process and yields 36 or 38 ATP. And you should also know that this respiration is a common process between plants as well as animals. So while the process of photosynthesis produces glucose in plants, the process of respiration is used to extract energy from carbohydrates, from glucose molecules in animals as well as plants. Now we should first understand the digestion and absorption of carbohydrates in case of animals. All the carbohydrates being consumed by animals are first digested in their alimentary canal. For example, in our alimentary canal, starch that we obtain from plants is digested in stages. First it is digested in our mouth by ptyalin, that is salivary amylase. Then it is digested by gastric amylase in the stomach, and then it is digested by pancreatic amylase in the small intestine. And once starch has been digested to smaller pieces, limit dextrins and trisaccharides or disaccharides, then they are digested by enzymes such as invertase, maltase, lactase or raffinase, etc. You know that invertase digests sucrose into glucose and fructose, maltase digests maltose into two glucose molecules, and lactase digests lactose into glucose and galactose. After digestion, you should know the mechanism of absorption of monosaccharides, which of course varies in case of different sugars, and there are two mechanisms for absorption of different monosaccharides from the luminal surface of mucosal cells. One mechanism is secondary active transport, which is applied in case of glucose and galactose absorption, whereas in case of fructose, the absorption is by facilitated diffusion. Now I wish to show you the details of both these mechanisms. Students, here I am going to describe the mechanism of absorption of glucose, galactose and fructose very briefly. But you should remember that this is an extremely important mechanism that you study in the physiology of digestion and absorption. Here you see that the absorption of glucose and galactose takes place by a similar mechanism, and it is called secondary active transport. Here you see that this is the section of intestine, and in that section of intestine the inner cell layer is called mucosa, and all monosaccharides are absorbed through these mucosal cells. So here I have enlarged these mucosal cells. Here you can see two mucosal cells in highly enlarged view. And as you can see that this face of the mucosal cell is called luminal face because it faces the lumen, and it is characterized by the presence of this brush border. This is brush border of villi. So toward the luminal face you will find brush border of villi, whereas the other face is called transluminal face. So students, this mechanism of glucose absorption or galactose absorption is extremely interesting, and it is called secondary active transport. Actually what happens is that on luminal phase glucose or galactose is always absorbed in collaboration with, in association with sodium ions. So it is necessary for glucose to be absorbed along with sodium ion, and that's why you might have noticed that whenever someone faces dehydration problem, then he is administered ORS. ORS contains salts along with glucose. So why glucose is given along with salts? The reason is that salts, means sodium ions, are necessary for absorption of glucose. So what happens that sodium along with glucose is transported through this molecule which is called sodium glucose linked transporter. Here you see this is called sodium glucose linked transporter or SGLT. So SGLT transports glucose along with sodium, and in this sodium moves from, sodium is high here, see sodium is high here and sodium is low here. So sodium is moving from, you see, sodium is moving from high concentration to low concentration. That means it is moving down its concentration gradient. Here you see that sodium is moving down its concentration gradient, and when something moves down its concentration gradient, such a movement is called passive movement. So here you see that sodium is moving passively and glucose is also moving along with it. So you might think that this movement is passive movement, but if you consider the other fact that why this sodium is low here, why is it that sodium is low here? So the reason is that there is another molecule on the transluminal phase that you are familiar with. This is called sodium potassium ATPase, or in short you call it sodium potassium pump. So I am writing it, this is sodium potassium ATPase. So sodium potassium ATPase or sodium potassium pump, what is it doing? It is doing, it is just binding 3 sodium on this side and 2 potassium on this side, and then it utilizes the energy of ATP, and utilizing that energy it rotates, and once it rotates, so it carries sodium from inside of this mucosal cell towards the transluminal side into the blood. So it transports sodium out of mucosa cell and brings potassium into the mucosa cell. So when it does so, what is it doing? It is taking sodium away from the mucosa cell. So that causes the sodium concentration to become low, and because sodium concentration is low, that's why sodium rushes from here down its concentration gradient. So now if you see both things together, so you see that this passive movement of glucose down its concentration gradient is made possible only when sodium is pumped actively out of mucosal cell. So this is active transport. This active transport is necessary for the functioning of this passive intake of glucose. So when there is such a mechanism where an active process drives a passive process, such a transport is called secondary active transport. So students, now you can understand why this process is called secondary active transport. So on this phase, on the luminal phase, so here I am talking about the transport of glucose on the luminal surface. So here you see that the transport of glucose, the intake of glucose on the luminal surface of mucosal cell, intestinal mucosa cell, is appearing to be passive, but it is driven by an active process taking place on the transluminal phase, so it is called secondary active transport. Now coming to the transport of this glucose or galactose across this transluminal phase. So you see that this implies different mechanism, and this is transported by molecules called GLUT2 molecules, GLUT2 transporters, which transport the glucose or galactose molecules which have already come inside the mucosal cell through a passive mechanism. So while the absorption of glucose on luminal surface is secondary active transport, on the transluminal surface it is simply facilitated diffusion through GLUT molecules. So this is all about the absorption of glucose and galactose. Now coming to the absorption of fructose molecules. So you should know that fructose absorption is different from glucose or galactose absorption, and all the way it is facilitated transport, and it takes place through a variant of GLUT molecules which are called GLUT5. So fructose is from the intestinal lumen, it is absorbed passively through GLUT5, and on the transluminal surface also it is absorbed through the same mechanism. Now students, once you know the differential roles of SGLTs, means sodium glucose link transporters, and GLUTs, means glucose transporters, in the transport of various monosaccharides, it is very useful for you to know their differential distribution. See that different variants of SGLT and GLUTs are found in different tissues of our body. Here I am presenting a summary of the main tissues where these different types are found. As you can see, SGLT is common on luminal surface of intestinal mucosa and cells of proximal state tubule of nephrons. Another variant of SGLT, that is SGLT2, is found in proximal convoluted tubule, which is again a part of neurons. And in GLUT molecules, there is a variety. GLUT1 are common in placenta, liver and blood-brain barrier. GLUT2 that I described with reference to intestinal mucosa cells are found in liver, pancreas and kidney also, apart from intestine. GLUT3 are specific for neurons in our central nervous system. GLUT4 are found in skeletal muscles and heart muscles and adipose tissues. And one important feature is that they are insulin dependent, whereas others are insulin independent. Now GLUT5 are again found in intestine, and their specific feature is that they allow the transport of fructose. Lastly, GLUT7 are found in the endoplasmic reticulum of liver cells, means hepatocytes. Students, finally let me summarize the various metabolic pathways of carbohydrate metabolism that I will be discussing in my future videos. So here is a quick summary. Here you see these are the major pathways, out of which first is glycogenesis, which is the formation of glycogen from glucose that takes place in liver. Whenever there is extra glucose in our blood, it is accumulated and stored in the form of glycogen. Then the reverse process of this is called glycogenolysis, in which excess glycogen is broken down into glucose. Here you see glucose changing to glycogen, glycogenesis, and the reverse process called glycogenolysis. Then there is very important process, glycolysis, which is ubiquitous and is found in all aerobic and anaerobic organisms, and it is the process used for harnessing energy from glucose. It results in production of pyruvic acid and NADH and ATPs from glucose molecule. It takes place in cytosol of eukaryotic cells and prokaryotic cells as well. The reverse process of this is called gluconeogenesis, in which glucose can be synthesized from proteins or proteins or lipids proteins or lipids may form pyruvate, and then pyruvate may form glucose in a reverse process of almost a reverse process of glycolysis. So this is called gluconeogenesis. Then the next pathway is called HMP shunt or hexose monophosphate shunt, and in this process incomplete oxidation of glucose takes place. There is incomplete combustion of glucose, in which 3 glucose molecules convert to 2.5 glucose molecules, which are restored, and half of glucose molecule is released as CO2. The importance of this pathway is the production of NADPH. So this pathway generates NADPH. From NADP changes to NADPH H+, which is important for oxidative metabolism of our cells. Then next important pathway is glucuronic acid pathway, in which glucose molecule forms UDP glucose, then that forms D-glucuronate, and then L-gulose, and finally it forms ascorbate. Ascorbate is what? This is vitamin C, and it also generates pentose sugars and some other important biomolecules like peptidoglycans, etc. Student, you should also remember that this step in which glucose changes to ascorbic acid or vitamin C, this step is lacking. The enzyme responsible for this step is lacking in humans. So this vitamin C cannot be synthesized through this pathway in our bodies, and that's why we have to take vitamin C from outside, and that's why we call it a vitamin in case of humans. But this is not so in case of many other animals. And lastly, sir, we have some miscellaneous pathways in which glucose may be interconverted to fructose or galactose, etc. So this is all about the overview of carbohydrate metabolism. In our future videos, we will be discussing each of these processes in full detail, and a separate video will be devoted to each of these processes. So please keep watching my videos and give your feedback also. Kindly share these videos.